Okay, this is going to be a series of videos I'm making to replace the face-to-face lecture that we missed on Wednesday. And I'm going to be talking about the material up on Blackboard. Lecture 5, Chapter 14, Innate Immune System 2 with PRR. So I'm going to be talking through the slides. I'll break the video up into sections and I'll post them individually. Okay, so we had just talked about the idea of pattern recognition receptors as part of the innate immune system. And one of the things that those, indeed the purpose of those PRRs is to detect stuff in the environment that tells our bodies a pathogen is inside us. So a pathogen is any microbe that can cause us harm. So there's these kind of good microbes that live on and in our body, we can call our normal microbiota, and then there's microbes that will harm us, and we call those harmful microbes pathogens. So we have these molecules called MAPS, M-A-M-P-S, and they stand for microbe-associated molecular patterns. And these are the things that tell us that microbes have invaded our barriers. So if, for example, we find peptidoglycan in our tissues, there's only one place that peptidoglycan came from. It came from an invading bacteria. Or if you find flagellin, the protein subunits that make up a flagella of a bacteria, then we know that there are bacteria in our tissues. And there's not supposed to be bacteria in our tissues, so if there is, we will mount an immune response. So the way we look out for these MAPs is through pattern recognition receptors. And on slide 1 there, we have the idea that there are two kinds of things we're looking for. Not just bacteria, but also looking out for viruses. And there are two places that we need to check outside of cells in what's called the extracellular environment, and we need to check inside cells in what's called the intracellular environment. On the outside of cells, we are looking for bacteria and viruses. On the insides of cells, looking for viruses in particular. Viruses have to get inside cells to replicate, but there are also many bacteria that want to get inside our cells. So we're looking for bacteria and viruses both outside the cells and inside the cells. So when we're looking on the outside of cells, we have what are called toll-like receptors. And toll-like receptors are anchored in the membranes of sentinel cells, there on slide 2. They can see MAPs in the extracellular environment. So we have, say, a macrophage. And the macrophage is just hanging around in our tissues. And it says there that our toll-like receptors are in what we call sentinel cells. Sentinel cells like macrophages and dendritic cells and mast cells. Cells in our tissues who are just part of the innate immune system looking out for invaders. So here's our macrophage, and there's a bacteria swimming around in our tissues, and that bacteria is shedding molecules. So parts of its cell wall, such as peptidoglycan, parts of its flagella, such as those flagellin molecules, parts of its membrane, and those are all then available for the macrophage here to detect. And it detects those with protein receptors embedded in its membranes. The other place we find these toll-like receptors are in these structures that we call phagosomes. So a cell like a macrophage is a phagocyte, and that just means a cell that can eat. So it'll detect this bacteria here, and it'll extend its pseudopods around the bacteria, and we have our toll-like receptors in the outer membrane. Eventually, the macrophage will completely surround the bacteria with a membrane. So not only do we have toll-like receptors facing out of the cell, we have toll-like receptors facing in at this bacteria within this membrane structure called a phagosome. So our macrophage eats by phagocytosis, and it puts the bacteria inside this little membrane bubble called a phagosome. It's going to inspect that bacteria. It's going to look at all its molecules using its toll-like receptors. If it's a dendritic cell, it would then transfer this information. It would crawl and it would reach the lymph nodes, and it would then talk to the cells of the adaptive immune system. So on to slide 3, we have rig-like and nod-like receptors. Rig-like receptors on slide 3. These are the receptors that are inside cells, and they are looking for bacteria and viruses that have got through the cells and or through the cell membranes and are living inside the cells. So rig-like receptors are picking up viruses, and one way you can try and remember it is rig has an R as I-R-I-G, and virus has an I, so rig detects virus. In particular, it is detecting viral RNA. So RNA, ribonucleic acid, an important kind of nucleic acid along with DNA. And viral RNA has some important differences. So your cells are full of RNA2, but there's important differences between the RNA of a virus and your RNA. For example, RNA of a virus doesn't have these caps, which you can read about on page 193. And often viral RNA is what we call double-stranded. Most of our RNA is single-stranded. So we have these receptors inside cells, and most cells have these rig-like receptors. So here's a regular cell, and there's our receptors floating around in the cytoplasm, and a virus gets into the cell. That virus makes RNA, and then the RNA is detected by our rig-like receptors, and these receptors then cause some kind of response. One important response of rig-like receptors is to trigger the cell to release this cytokine called interferon. What interferon does is it gets the cells around to prepare themselves for a viral infection. So our cell here in the middle has been infected with the virus. Here are all our cells in our tissues surrounding that. So our cell here in the middle has been infected by a virus. The rig-like receptors have detected that virus, and the cell has been then releasing interferon. And that interferon is picked up by the surrounding cells and they are ready to go. Then if the virus gets out of our cell and it wants to then get into the neighboring cells, our neighboring cells will self-destruct as soon as that virus gets inside. They are ready to detect the viral activity. The virus gets inside and the cells go through apoptosis. So the interferon has caused the surrounding cells to produce these suicide proteins that are watching out for viral RNA, and as soon as they see it, the cells kill themselves. And it's like a firewall in that these cells have killed themselves before the virus can continue to spread. So the rest of our cells are saved. On to slide 4 is nod-like receptors. And nod-like receptors are again inside cells. They are particularly important, as it says here, in macrophages and dendritic cells. They are detecting bacteria that have got inside our cells. They're also detecting cell damage. So when a cell gets damaged, it'll make and release all kinds of chemicals. The inflammasome is a product made by macrophages and dendritic cells that join together and produce this kind of immune complex called an inflammasome. But we aren't going to talk too much about that. Okay, this is the first section. Number 5, slide 5 has the study guide points. So I'll end this first video right there.